Good day and welcome, everybody. It's with great pleasure to see so many of you already, and I think we will get a few more to the 29th session of the Global Ocean Oxygen Network webinar series. So 29, we didn't make it 30 yet, but soon to come. Happy belated World Ocean Day. I'm Karin Limburg from the Ocean Science section at the Intergovernmental Oceanographic Commission of UNESCO, and together with Jeremy Sterling here as well online, we provide the technical support to the Global Ocean Oxygen Network and the Global Ocean Oxygen Decade. It's a very special GO2NE webinar, as we are quite lucky to have here with us as well the two co-chairs of the GO2NE Expert Working Group and the long-standing serving member as well of GO2NE. We will have 3 talks in total. Each speaker will have around 20 minutes, and we will have time for 2 question and answer sessions. 2 speakers, so Caroline and Karin will talk in the beginning, and we have some question and answers, and then as well Andreas. To put your question and answers, we ask you please to put these in the Q&A box, which you should find on the Zoom panel there. And the chat box is also, of course, open, and you can put additional information in there and to exchange with the audience. And before I hand over to our first speaker and for Caroline Slomp, and then over to Karin Limburg, I would like to give you a very brief overview of the World Oceans Day. That's why we are here. This was actually instituted under the United Nations in 1992 with the aim to inform the public of the impact of human actions on the ocean, develop a worldwide movement of citizens of the ocean, and mobilize and unite the world's population on a project for the sustainable management of the ocean. So everything we are concerned with, especially as well within group, falls into that, so the loss of oxygen. The GO2NE and its members support this by participating in events and providing messaging on ocean oxygen to existing initiatives. This year, and also last year already, members did and will still participate in many in-person and online events, ranging from lectures to demonstrations to meet the science in school events and many other outreach activities. You can find much more about that and other activities of the GO2NE working group and the Good Decade program on the link that you will see at the end of the webinar, Ocean Oxygen. Is it called. So, but I won't take too much time here, and really let's go to the important stuff, and I'm very happy to introduce Caroline Slomp, as mentioned, co-chair of the Global Ocean Oxygen Network, and she will provide us some more insights on the global and regional examples of ocean deoxygenation. Caroline, the floor is yours.
Okay, thank you. I'm going to share my screen. Okay, so hope you all can see it. Thank you very much for this invitation to talk here today. And as just mentioned by Kristen,
sorry,
I'll be
Carolina.
Okay,
I'll just cut in quickly. We can't quite see your screen yet.
Oh, you can't? Okay. How's that possible? I'll go back. Oh, wait a minute. Leave presenter. I'll try this again. Sure.
Perfect.
Now you can. Okay, sorry.
Thanks so much.
Okay, let me start again. So, well, it's a great pleasure to speak here today about ocean deoxygenation. I'll be giving a short introduction on the drivers of ocean deoxygenation, on its impact, but I'll also spend some time on solutions, and those will be regional examples. This is a very well-known figure from a paper by Denise Breitberg and others from a couple of years ago, summarizing let's say, the changes that we're seeing in the coastal and open ocean waters linked to low oxygen. And the red dots indicate coastal areas that are subject to a decrease in oxygen or are suffering from low oxygen. And the blue areas are the oxygen minimum zones that we know have been expanding in quite some areas in vertical extent, but also horizontal extent. And here we define low oxygen basically when concentrations are below 2 milligrams per liter. And anoxic refers to when you have no oxygen in the water. Now, we know why we have this low oxygen. Low oxygen in seawater occurs when its removal outpaces its supply. And we know the supply terms. We know there is exchange of oxygen between the water and the atmosphere. We know there can be physical transport, which can be lateral or vertical, that supplies oxygen. We also know that oxygen can be produced in surface waters upon primary production. And then removal occurs through respiration of the organic matter that is produced. And that can occur in the water column, but also in the sediment. And we also have reduced components that can be formed in sediments during organic matter degradation that can later remove oxygen. And an example of that is, for example, hydrogen sulfide. Now we know that the oxygen in the ocean is decreasing because of warming, and we also know that eutrophication is increasing the oxygen demand. Now to discuss those two mechanisms in a let's say more in the framework of the ocean, it's important to realize that eutrophication is basically impacting the coastal ocean. And so eutrophication, so that means the leakage of nutrients, especially nitrogen and phosphorus from land into the ocean, and that fuels algal blooms. And when these die and sink to the seafloor, they can remove oxygen. And they also can remove it, of course, during sinking in the water column. And this is why in a lot of coastal areas, we are seeing low oxygen emerging. Now, in the open ocean, the major driver of deoxygenation is climatic warming. And because of warming, you can get less dissolved oxygen, and also the increased stratification linked to global warming leads to reduced mixing, so less ventilation of deeper waters. And so in oxygen minimum zones, we see the expansion basically due to global warming. We see this, as I mentioned already, a horizontal and a vertical expansion. Now, most of my talk will focus on coastal areas, so I put in this slide just to emphasize that these major changes that we're seeing in coastal waters are mainly occurring since around 1950. This is when human population started to increase dramatically, and this is from a paper by Nancy Rabelais from 2014. You see the very strong rise in human population, but also associated with that, a very strong increase in fertilizer use. And the yellow line here is the use of nitrogen fertilizer. And what we also know is that in developing countries, this occurred later than in developed countries. So we also see a shift in places where hypoxia is emerging. And we see that in a lot of countries, people are also taking measures, and I'll come back to that later. Now, this low oxygen has dramatic impacts on coastal and open ocean systems. It impacts marine life. And this is a schematic from a very important and useful report, IUCN report on ocean deoxygenation that you can find on the web. And a lot of GO2NE members contributed to that. And what this figure shows is that, in fact, if you have oxygen saturation, then fauna is okay. But as soon as your oxygen concentrations go below that 2 milligrams per liter that I mentioned before, you see fauna disappearing, especially fish will be absent. You'll have shrimp and crabs disappearing, and especially when you have low oxygen or no oxygen, and when hydrogen sulfide builds up in the water column, no macrofauna will survive. So you see this whole gradient. There are other trends in between that don't have time to discuss, but basically you see a decrease in fauna when you have low oxygen. And this also plays out in coastal systems. You see fish kills, you see fish getting trapped. and dying. You see sometimes even fish trying to crawl onto land, and crabs crawling onto land and dying. And this is a picture taken not long ago of dead crabs, where there was a clear link with a low oxygen event near the coast of South Africa. So we know that low oxygen is increasing. We know that we know the causes, and we know that it has major impacts. But not only on fauna, it also has impacts on microbes. And what we see is that a lot of areas actually turn into, let's say, that they get covered with microbial mats. We don't like microbes, microbial mats when they're visible. And this is the location in a Dutch lake where divers like to go for recreation, but it becomes a very unattractive place if you have all these microbial mats, these sulfur oxidizers. These are, in fact, so these are microbes that live off of the sulfide that is seeping out of the sediment. Now, we know what to do about this. We know what measures to take to reduce ocean deoxygenation. And it's pretty simple. It's basically reduce greenhouse gas emissions, so carbon dioxide, methane, and nitrous oxide, but also reduce nutrient inputs to coastal environments. Now, even though we know what to do, we also know that it is difficult to achieve these changes both on a national and a global scale at present. And this is why a lot of ideas have developed about how to maybe quicken or speed up that process, and also why people are really very closely tracking the impact of local measures to reduce nutrient inputs. And I want to discuss 3 examples here now. And these are 2 large systems, the Baltic Sea and Gulf of Mexico, but also Dutch coastal waters. And the Baltic Sea is known to be the world's largest anthropogenic dead zone. And on the left-hand side, you see what the Baltic Sea, let's say the oxygen state in the Baltic Sea more than a century ago, and the red colors and black colors indicate where the waters below 70 meters were were either euxinic, so containing hydrogen sulfide, or hypoxic, so below that 2 milligram per liter. And what you can see is that there was very little hypoxia and euxinia in the Baltic Sea around 1906. But if you look at the year 2012, and this continues to this day, you see that there is a massive area, a huge area of hydrogen sulfide rich water, and also hypoxic water. And this is from a paper by Jacob Carstensen from 2014, but this still holds today. And we know that eutrophication is the key driver here, so nitrogen and phosphorus input. And we also know that the Baltic Sea is very sensitive to low oxygen, so it's easy to tip it into this low oxygen state. And this has to do with its geography and its limited connection to the North Sea, and also the associated long residence time of the water and the nutrients. So people discovered this quite some time ago, that this problem was existing. So since 1980, nutrient loads have been reduced quite a bit. But if you actually look at the hypoxic area, so the total area of low oxygen and also sulfidic bottom waters, if you take that together and look at that, you see that it has expanded greatly since 1900. There's some gaps in the data, but still there's a general increase. There's also some fluctuation due to weather conditions, but what we also see is that there is no recovery yet. So we're already 45 years past the start of the reductions, but there's no recovery yet. Now we know that there are processes that keep systems in a low oxygen state. So apart from the residence time, you also have other processes that can make it more difficult for systems to recover. And I'm going to highlight 2 of those processes here. And one is on the left-hand side of this slide. One is related to the fact that one of the 2 nutrients that I've been mentioning, so nitrogen and phosphorus, one of the 2, that's phosphate, becomes more efficiently recycled in the system when a system becomes low in oxygen. So what this means is that phosphate can be used again and again to produce organic matter because it simply is not lost easily from the system. And in this case, that would mean burial in the sediment. So what you see is that the phosphate can fuel, especially cyanobacterial blooms, where the cyanobacteria gets their nitrogen from the atmosphere. That leads to more hypoxia, and that feedback loop is keeping the Baltic Sea in its low oxygen state, or at least that's one of the factors. And another important factor is indicated on the right-hand side. That is that you have this massive deposit in the Baltic Sea now of organic rich sediment that is releasing sulfide, ammonium, and methane, so products of the anaerobic degradation of organic matter. And if you try to reoxygenate the system, actually that oxygen is removed because of this flux of reductants from below. And so this means that it's very difficult to actually change the system, flip the system back. And this is something that we showed a couple of years ago. Now, what can you do to improve water quality in the Baltic Sea? What are there options apart from the measures mentioned already? Are there options to quicken this? And so there have been quite some proposed quick measures like pumping oxygen into the deeper water, increasing water column mixing, also by pumps or by windmills, and also by precipitating the phosphate with chemicals. Now, there's a nice review about this. It's now 15 years old, but a lot of the conclusions still hold, that it's actually quite problematic to do this on this large scale, basically because the scale is so large. So it's really hard to do enough to actually impact this system. Because remember, this is a huge low oxygen system. And then the biogeochemical feedbacks can mean that you can put in a lot of effort without getting a result. And some of these biogeochemical feedbacks also could mean that if you start pumping, you have to keep on pumping. Otherwise, you might flip back to a state that could be worse. And we know this from a lot of lake studies. Because you could create a pool of phosphate that is can be remobilized, and you can enlarge the current pool of phosphate. And then there are a lot of unknown biological effects. And so, and there are ethical considerations and legal considerations, of course. I don't have time to go into all the details here, but one of the, basically for this system, you could say a good solution is actually to address global warming. Put your euros in addressing global warming and reducing nutrient inputs and be patient. Now, what about the other major coastal human-induced dead zone, the Gulf of Mexico? That area receives a comparably high nitrogen and phosphorus load to the Baltic Sea, and also has a very large area of seafloor that's low in oxygen. The Baltic Sea is an area of about 70,000 square kilometers. Gulf of Mexico, 1/3, but still that's an enormous area. Another difference is that the Baltic Sea hypoxia is near permanent and only has occasional, let's say, inflows of North Sea water that occasionally briefly reventilate. The Gulf of Mexico hypoxia is seasonal. And so it's a very, very different system. Now, what's important there as well is that the Gulf of Mexico dead zone has a much shorter residence time. And I already said the Baltic Sea has this long residence time. It's actually on a timescale of decades. And here the residence time is only a month. So that's a really big difference. So this also means that this is a system where there's more potential for fast recovery. And another really nice finding is, in fact, that the sediment feedbacks in this system are more limited. And so this is work that various people have shown this, and that basically there's less sulfide and less methane coming from the sediment seeping upwards and removing oxygen and ammonium, I should mention as well. So there are much less biogeochemical sediment feedbacks. There's less phosphate recycling, and so basically, this system is easier from the seaward side, is easier to recover, or can be easily recovered. even though climate change could intensify the anoxia and extend its duration in the system. But where the problem lies here is, in fact, that you have a very large river basin, and there's a lot of legacy nitrogen in this basin that will continue to wash out even if you would stop or change management practices. So here the legacy problem mainly lies on land. Now, what about the recovery of smaller coastal systems? And of course, there are many, many of those small coastal systems. So I'm from the Netherlands, and we're doing a lot of work on this in Dutch coastal waters. So I took let's say, a nearby example to discuss with you today. And in the Netherlands, there are a lot of estuaries that were dammed in the 1960s and 1970s because we had a massive flooding in the 1950s, and people said, never again, we want safety. So dams were placed, and these former estuaries were turned into seasonally anoxic marine lakes. And they knew this was going to happen, but still the water quality problems are large. And one example is Lake Vere, which is a former estuary that developed strong bottom water hypoxia. So it was dammed on two sides in the early 1960s. And you can see from the map on the left-hand side that this lake is a very long lake, and it's basically, as you see, an estuary dammed on two sides. And so at some point, the conditions became so bad that they decided to open up a sluice in the dam at the eastern side of the lake to increase the water exchange. And so this initially, and you can see the sluice on the right-hand side. And so what this basically did was bring in water into this lake and increase its oxygen supply, increase let's say the lateral and vertical water transport. So here you can see that on a smaller scale, you actually can take measures to improve the oxygen conditions. Now, this happened in 2004. Recently, a severe decline in water quality in the lake was discovered. And on the left-hand side, you can see the lake. You see stations where samples were taken. And you can also see above, you can see contour plots showing with red colors the low oxygen. So the hypoxia has returned, but now mainly on the western side of the lake, and also the middle part of the lake. And people observe fish kills, a lot of jellyfish, a lot of microbial mats, and it's become an unpleasant place to be. And so you can see restoration can work for a while, but eventually can fail. And so the question then is, why was the restoration not more successful? What didn't help in assessing that question, or doesn't help, is that the monitoring was also reduced because people thought it was a success. But our current hypotheses are that the increased input of organic matter from the adjacent marine system actually has an effect on the oxygen budget of the lake, and also that there's an effect of warming, because our summers are becoming warmer, and so the world is changing around us. So what is needed on a short term is further nutrient reductions and increased water exchange, and if possible, also a reduction of organic matter input from the adjacent system. So just to wrap up here, so the take-home messages here are that really, if you want to effectively change low oxygen, you need to limit, or the expansion of low oxygen, you need to limit global warming, you need to reduce nitrogen and phosphorus inputs. And that you can have all sorts of feedbacks that can play a role, but they don't always. So that's also helpful. So you also need to look at things case by case. So biogeochemical feedbacks are not equally strong in all systems. And that sometimes you think you found a solution to low oxygen, but it might not necessarily last, and you might create new problems. So it means you need a tailor-made approach for each system. So there, the regional side is really, really important. And so with that, I'd like to thank you for your attention, and I'm going to be handing over to Karin, who will continue.
Okay, thank you. And I think you need to stop sharing your screen. Okay. I just want people to see that I'm wearing my hypoxia shirt today. And it's wonderful to see so many people from so many parts of the world, I have to say. Now I'm going to go ahead and show you my screen and get to the right place. And there we go. And let me hide the meeting controls so I can see. And yeah, so let me get started here. I've been asked to address the some of these biological and ecological impacts of deoxygenation, and I also wanted to mention that a group of us in the GO2NE, led by Mike Roman and Lisa Levin, have a manuscript that's in review currently in Biogeosciences that is a pretty extensive review of things. And I will give kind of a fish-centric presentation today because I'm a fisheries ecologist. But these kinds of things that I'm going to talk about extend across most animals in water. So I like to think of this kind of, I'm giving you kind of a scaled perspective from thinking about energy budgets, which would actually take place from cells all the way up to ecosystems, but kind of I'll focus kind of on the organism, the physiology too, touch on sensory systems, touch on the immune system, and growth and condition, population effects, which I've done a lot of work on, habitat use and food web structure and ecological interactions. I won't really get into economic implications so much, but we might take that up in the Q&A. So with the energy budget, this is a slide I actually show in my class, and this is kind of like the basic, you know, there's an organism eats food, it excretes, it respires, metabolizes, and what's not lost from those processes goes into growth and reproduction. And aerobic metabolism is the process that yields the most energy. It's about 6 times more efficient than anaerobic. It's a very, very large yield in ATP. The physiological impacts of low oxygen, you know, if we want to address that, aquatic animals require oxygen to power their life, and reducing it leads to a cascade of impacts that I'm starting to think of as kind of a hypoxia stress syndrome. And I want to also just mention quickly that all of these things, although we're observing World Ocean Day today, this is also happening worldwide in inland waters as well, lakes and rivers and so on. But you see a cascade of effects: slow digestion, poor avoidance of predators, harder to find your food as well, immune responses, which can be increased susceptibility to infections and parasites, and morbidity, and then death, of course, as Carolina showed in extreme cases. But it's not just it's not just plain old oxygen concentration, it's the percent of oxygen that we care about, because as that partial pressure drops, so does the ability of oxygen to get into the tissues where it needs to be. And that can be calculated. That's, if any of you have used an oxygen meter, you'll know that percent saturation is typically reported, and that is a function also of temperature and salinity. And I'm sorry that this is a very complicated looking graph, but it does kind of have everything on it I wanted. What we have here is a graph that plots metabolic activity against dissolved oxygen as percent saturation, as I said. And this upper line here, this curve, represents the maximum activity, basically metabolic activity with various terms on it, maximum oxygen uptake and so on. And this blue line at lower down is the standard metabolic rate, which just basically meets all the needs of the organism when it's not doing anything else. And the difference between these two lines is called the aerobic scope. And as you go down in oxygen saturation, you kind of go down in your scope too, because the standard metabolic rate is fairly constant. And then you eventually can get down to a level that's called a critical oxygen level where you can't really do anything. But this also shows how various processes are limited by reduced oxygen. So digestion goes down, ability to reproduce goes down, and so on. And then you can get into a point that's very dangerous of accumulated oxygen deficit, and then, of course, death as well. But a different perspective on this is looking at it as a function of temperature, which is the way I was more trained to do. And when you look at metabolic rate as a function of temperature, there's typically an optimum temperature for your metabolic rate, and then that declines as you get beyond that optimum, and as your proteins start to deaminate, you'll eventually die, but it will meet with the standard metabolic rate. So that will go up exponentially as a function of temperature. And so if you lower the DO, you can see where this would be going, that you would be lowering and lowering that. So temperature is very important as well. Of course, there are some animals that are actually super tolerant of low oxygen levels. They are certainly in the minority, but I just wanted to let you know that there are a few. One of the most super tolerant is this little creature here on the right, the bearded goby from the Benguela ecosystem off of Namibia, and it can actually tolerate not only anoxia for periods of time, but even some hydrogen sulfide, it turns out. But it's a very unusual animal. So if we touch on sensory systems, a very nice review by Lily McCormick and Lisa Levin that I recommend reminds us that vision is very oxygen demanding, and that neurons and photoreceptors are very, very energy demanding and oxygen demanding accordingly. They metabolize aerobically, and that we know of cases of visual hypoxia that can affect things like tunas and sharks and cephalopods and some arthropods too. Hearing is also affected. There's documented by experimental observations temporary loss of hearing in anoxia, and also loss of a fast startle response. And that will have ecological implications because of increased vulnerability and just things like decreased foraging ability and so on, and possibly also loss of communication, because we now know that fishes and other organisms are actually communicating with sound. The image on the right is a image of two fish ear stones, or otoliths, from cod that I've been studying. And this refers to this sort of longer term possible impact, which is that something about the structure of these organs. These are calcium carbonate structures that grow throughout life, and as a fish gets older and older, they get more sculpted and more ornate. And what I noticed in the Baltic Sea cod, which we'll get to in a minute, they're very, very smooth now, and this does seem to be affecting their ability to vibrate properly and move properly under different sound conditions. There's also hormonal systems that we know are very well documented with deoxygenation, that the stress hormone cortisol levels increase, growth factor goes down. We're also looking at that, reducing immune function and feeding suppression. And then there's been a well-documented case in the Gulf of Mexico of the Atlantic croaker, this fish down at the bottom, showing a reduction in reproductive organs, a dramatic change in size and function of those. I also wanted to mention behavioral effects, and there's a great example using billfishes, so the large predatory fishes, the tunas and billfishes and so on. And this study was done on billfishes in sort of the Caribbean and the other side of the peninsula here, looking at hypoxic billfishes in hypoxia versus normoxia. putting satellite tags, pop-up satellite tags on them. And what we can see is that on the Atlantic side, colored in red on the right-hand graph, we can see that the billfishes can dive deeper and stay longer down at deeper depths than they can in the Pacific side. And this Pacific side phenomenon is called shallowing or habitat compression, and we know that this affects their ability to forage greatly. I'm going to give you a quick case study of what I've been working on. I have a project that has been nicknamed Project Breathless, and we're looking at the consequences of hypoxia in the Baltic, Lake Erie in North America, and the Gulf of Mexico in North America. But I focus on the Baltic, and this is just a very dramatic story, which is that cod have been very, very severely hurt by hypoxia, and this is the map that Carolina showed before. But what I want to show here is that the low oxygen has greatly reduced the ability of the fish to spawn. So they need a certain amount of oxygen at depth to be able to spawn because of the salinity requirements that they have. And so now there's a greatly reduced spawning volume. There's also this is perhaps a bit of an exaggeration in the 80s, but there were very, very strong year classes with large fish with many age classes. And today they are definitely very much smaller. And it's the incidence of these, what the Swedes call necktie cod, very starved, has increased as well. So recognition goes down. The chemistry of these otoliths, as I mentioned before, the chemistry can be used to track exposure to hypoxia. We can take one of these otoliths and we can section it, and then look at that section. We can see the age rings throughout life, but we can also see manganese with different instrumentation, and this was done with a synchrotron. And manganese goes into solution at very, very, very low oxygen levels, and then kind of persists for a little bit, but that's enough for it to get taken up into the earstone. So we can see the high levels during summertime exposure to hypoxia. And what we were able to do was to see that we could develop a duration proxy of it. We looked at intensity, exposure, and duration. And what we found, sort of surprisingly, was that this matched up extremely well to an oceanographic index that's calculated every year for the hypoxia and anoxia volume of the Baltic Sea. So I was pretty amazed at that. But that does, it still persists. But what we could also do then was, another thing that fisheries people do is we can back calculate the length and age of a fish. And we could split the oxygen classes into, by quartiles, into least. The blue circles here are the least exposed to hypoxia, and the purple triangles are the most exposed to hypoxia. And what we can see is that the differences aren't so great when they're young, but as they get older and older, those differences do magnify. And so this is a measurement of exposure directly, as I say, from the heads of fishes. And by the time they're 3 years old, they're 39% smaller. And this actually translates into a 64% decline in weight. So that would mean then that you are losing biomass in hypoxia. It just doesn't develop. And so there are economic implications for that we're trying to work on. Another trace element, magnesium, reflects growth and metabolism. And one thing that popped out of our study, surprisingly, was a great difference between condition factor in fishes reflected in this magnesium. And it seems like this low condition does sort of persist over time. And then this this graph comes from a very, very recent report that just came out from ICES about cod in the Baltic Sea. And it shows that despite the fishery being closed in 2019, there was a great deal of controversy as to whether or not this would be the effect. Despite that, the population has declined greatly. Quickly through community or ecosystem level effects, the study we did of a hypoxic site outside of the Stockamar archipelago and then a normoxic site. What we see with some stable isotope approach is that stable isotopes will give you with carbon some idea of source, and with nitrogen some idea of trophic level typically. And what we see for all of these is that the corresponding green in the hypoxic zone fishes are all sort of pushed to the upper left compared to the blue and the yellow fishes. So there's some kind of shift going on in hypoxia. And that is actually seen as well in normoxia. When we expand the food web out just a bit more, we can see the same sort of trend, and this suggests that there's some denitrification going on. I'm very close to being done. So we can also examine this diet. We can examine the hypoxia impact on niche breath with a technique that's called Bayesian ellipse analysis. And so this puts statistically standardized ellipses around a field of data, stable isotope data. And what we see is that in normoxia, this particular fish, which is a flounder, has a larger envelope of diet reflected than a hypoxic one. And then this can be statistically quantified as well. And so this Baltic flounder are statistically very different in hypoxia versus normoxia. We don't, oddly enough though, we know that these system level effects could be different in different systems. And in fact, we're finding across the board that it's quite different in the Gulf of Mexico. We don't know why. But I think that this gives you kind of a quick overview of the fact that hypoxia effects can occur from the cells to ecosystems and affecting organisms and the ecology in many, many ways. And I didn't cover toxicity impacts here, but those are also another aspect. And that work remains to assess really the full economic costs, which in the short term can be positive, but in the long term, probably not. And so that is my presentation, and comments are certainly welcome. And I'll stop there. Thank you very much.
Okay, thank you, Karin. I'll hand over to Andreas Oschlies, who will be moderating the question and answers.
Thank you very much, both speakers, Carolyn and Karin. Very exciting presentations, and there's a couple of questions here. First, maybe to Carolyn. Are there any models to predict or detect hypoxic or anoxic conditions?
Well, basically, that's something that you will be talking about, Andreas, right? And so basically, there are regional models. Yeah, they're global models, and that's what Andreas will be talking about. But there are also regional models to predict this. So specifically for the Baltic Sea, there are multiple models that people use, and also people compare. So those are really important because models have advantages and disadvantages. The different ones, and then you can compare them and see where you end up and how the results compare.
Have they been successful in the past? So you showed this example for the Baltic Sea where oxygen declines despite additional nutrient input. Was that predicted by the models?
Yes, they predicted that. So they predict a delay in the recovery. And so they also, and that's the good news, they also predict a recovery. If you continue to reduce nutrients, then the Baltic Sea will recover. So it shows that that is the way to go, in fact.
Right, thank you. And then, a related question there is, could remote sensing be helpful to detect or the absence or presence of hypoxia or anoxia?
Basically, I know that remote sensing is used a lot to look at the cyanobacterial blooms in the Baltic Sea, for example, and look at primary. I mean, you can use satellites to look at primary production and to quantify that. For deoxygenation, I have not. It's deeper water, so it's not like something you could detect through remote sensing. So you really need to have sensors in place to actually detect low oxygen.
Thank you. And then what about hydrogen sulfide? Could that change the color, or is it quantitatively okay?
Again, mostly that's in the deeper water. You wouldn't see it. I know there are you sometimes can get these white precipitates of sulfur in the water. That would be something that you might be able to detect. But those are not common events. So it's not like there is a technique to actually map this from space. There is not.
Okay. Yeah, thank you. Then a question to Karin. What do fish do to tolerate anoxic or anoxic conditions, or conditions where even hydrogen sulfide is present?
That's a great question. That's I believe they just have they have they undergo anaerobic metabolism instead of aerobic, but I can't get you into the details of it. But there are papers. There was a nice paper written that appeared in a special issue of Frontiers in Marine Science in 2018 that I would recommend looking at it. So I forget the title of it right now, but this is about the bearded goby. The bearded goby is a very unusual fish because not only can it forage on bottom sediments in the daytime in anoxia, but at night it goes up into the surface waters to basically digest, I guess, and it hides amongst the tentacles of man-of-war jellyfish, which are deadly to most things. So they're really quite super tolerant in many ways. Other organisms that cope with low oxygen include some of the cephalopods, which are able to, for example, squids and octopuses, which are able to store a lot of oxygen in their tissue.
Okay, thank you. Then a question to both speakers. Do you think that 2 milligrams per liter is too low for a definition of hypoxic water, considering that some, well, sublethal effects may be expressed already at 3 milligrams or even higher concentrations?
You want to take it, Carolina?
Well, I think we basically, we cannot get a measure that works for all fauna, all animals. So I think we basically just need a threshold to use and to map and to be able to quantify things. But I think we're aware that it doesn't always hold. But this is my...
Oh, go ahead.
Yeah. No, go ahead. Here, Karin.
I was just going to mention that in Long Island Sound, in near part of New York State and Connecticut, that it's that the threshold that they use there is 5 milligrams per liter, so it's quite a bit higher. And in the Baltic Sea for cod, anyway, they sort of think about 1.5 milliliters per liter as the threshold for cod, which is kind of amazingly low in my opinion. But that's sort of an extreme threshold, I think, for that species. And then we know that many flounders, many of the fishes and animals that do live on the bottom or in the sediments are somewhat hypoxia tolerant, but not necessarily for prolonged periods of time. So we know that things like isopods, which can be very hypoxia tolerant, do eventually die out if it persists, if anoxia persists. And then we lose basic parts of the food web, which are very important.
Yeah, thank you. Would some metric like we have for coral bleaching, warming days, be possible for deoxygenation for anoxic days or something? Is that a useful metric?
It's a great idea. It's a great idea.
Then a question here is there any, well, there are specific time periods during the year at which suboxia or deoxygenation events occur. Are there specific times? And should institutions use different thresholds or different indicators at different seasons?
Well, we know that most hypoxia and anoxia occurs in summer. That's when you have most primary production and yeah, stratification of the water column due to temperature. And so yes, we know there's a seasonality there. About seasonal
Indicators.
indicators for biology, that's not a question I can answer. That's for Karin.
Well, I think again, it's you know, you can look at things like diel metabolism at the system level and look at changes in oxygen, but the biotic indicators, there are some, I don't have it at my fingertips, but there are some short-term markers of hypoxic stress that have been documented in fishes in the Gulf of Mexico too. So there are some biomarkers that are short-term and then some that are long-term. The kind of thing that I gave the example of manganese is one. I didn't get into, there's a huge field of indicators of hypoxia that range from another kind of tracer in otoliths, looking at oxygen stable isotopes. No, I'm sorry, carbon stable isotopes, and tying that to models that are called metabolic index models too, which look at basically the ratio of supply to oxygen supply available in the water to the demand of the organisms too. And that would be species by species, really, or taxon by taxon.
Yes, thank you. And then maybe a last question here. I think it goes to Carolyn. Can you comment a bit on how the problem is manifesting in developing countries? And is it growing as they grow their economies and coastal cities, as shown on one of your slides?
I think it is growing, and but we don't have a full overview of that because our monitoring and our measurements are mostly done, you know, in other areas. So this is something where actually we need more measuring effort. And this is also why a database of oxygen, which is one of the things that GO2NE and GO2NE is working on, is very important to have an overview of the data that are available. So, so yes, it is growing, and yeah, we need to know more about it.
Okay, thank you. Thank you very much, both speakers, and to all those who posed questions. I hope I picked those that are the different ones, and the others, please send us emails if you think they are not answered, if I missed some. Thank you very much. So that's the first part of this webinar.
Yeah, so then it's a great pleasure to introduce you as a speaker, Andreas, and you'll be talking on the state of global deoxygenation and the work of GO2NE and GO2NE. So the floor is yours.
Yes, thank you. So is this the right way? Or do I have to stop? It's okay?
Yeah, we can see it.
Good, thanks. So this is gives you the global overview, the global situation, and also the global call for action. And I think it affects all of us eventually. So we all are will be hit by deoxygenation. This is first a map of oxygen loss in the ocean over the past 50 years, so 1960 to 2010, compiled from all available observation during that time. Red is showing vertically integrated decline of oxygen. So we see a decline almost everywhere in the global ocean. And during those 50 years, there was a 2% decline. It sounds not a lot, but if you extrapolate it, the ocean would be devoid of any oxygen within 2,500 years. So this is really alarming. And at that time, 2017, this paper really changed our view of stresses in the ocean. We have looked at warming, acidification, but deoxygenation really came up into our view directly via this figure, this paper. And we have seen it in many locations before, but a global issue is really for the first time shown here. The question then arises, how will this go on? How will it continue into the future? Are we already on the edge of anoxia? So extrapolating this over civilization periods, we could easily imagine that if such a trend continues, we turn, well, into another mass extinction event. And that is what we think has happened several times on Earth in the past, that the oceans or large parts of the ocean, anti-ocean basins were anoxic. So that's where we have black shales or many of our current fossil fuels. And in the last 500 million years, we think there's a lead for these major extinction events linked to ocean anoxia. Maybe not all, particularly not the last one that killed the dinosaurs, where we have another mechanism, the asteroid, but all the others may be linked to anoxia, and many have been shown that are entirely linked to anoxia. So this is, of course, if we extrapolate a major reason for concern, and we have to try to look into the future. How do we do this? Well, we run models, climate models. That's what the IPCC does for global warming, for the air temperature, precipitation changes. That's what human concerns directly, but these models also have marine biogeochemistry. And so one can look at oxygen in these models. And that's done here for the most recent IPCC models and of the most recent assessment. And all these models show that deoxygenation declines over the next, well, this century. And it declines faster and more into low oxygen values if it gets warmer. So warmer oceans and a warmer world will have increased deoxygenation, reduced oxygen levels in the ocean. So this is what all current models predict. And again, it shows if we stop warming, we may stop deoxygenation at a less severe value. But still, even for 2 to 3 degrees Celsius temperature targets, if they are met in these model simulations, we still see a substantial decline of ocean oxygen levels. And this is one study where we turned off all emissions in year 2020 already. So that's what we call committed deoxygenation. What would happen if we stop warming today, if we stop emitting all CO2, burning fossil fuels, cutting down forests? What would happen today if we behave as we should behave from a climate perspective, at least? So this is zero emissions starting in 2020 and then continuing for 1,000 years in the simulations. And we see that surface air temperatures are stable as predicted by almost all models, so that we stop global warming, we have decreasing atmospheric CO2 level, the black curve, and we have an increasing ocean heat content. So the ocean basically takes up heat in order to balance atmospheric temperatures. However, oxygen in the ocean, the blue curve on this slide, continues to decline despite zero emissions. So in the zero emission world, all climate targets are met, atmospheric temperatures are stabilized, politicians are happy, society might be happy, and we have fulfilled all our promises. We still see a substantial decline in ocean oxygen. In the end, in these model simulations, it's a 10% loss, so much more than the 2% we've seen so far. And in fact, only 25% of all the committed ocean deoxygenation has been realized by today. 3/4 are still in the pipeline, even if we would stop emissions today, according to this model. And we don't know if models are good or bad. We mostly know they're not so good, but that's the only tool we have to look into the future. And even here, we see some surprises. After several hundred years, the ocean circulation rearranges. It reventilates the deep ocean, so it might lead to oxygen again, but that's not, of course, not enough for those organisms or species that died out. So holding your breath for several hundred years is very, very difficult and doesn't help. So what do we need to better understand, have more precise mechanistic understanding, know if our models are good or bad, and how reliable all these predictions into the futures are? We need data. And we have a lot of data, historical data first. So oxygen is the longest record of direct climate-related measurements. So it has been measured for more than 100 years by the same method, Winkler titration, that's developed 1888 and as in Winkler's PhD thesis. So very, very famous work. Any current PhD student and I would be happy if any of my work is cited in 100 years or more than 100 years, 150 now. So the same method has been used for more than 100 years. And so we have the same precision and we can directly compare measurements. And here's one example of the first ocean basin meteor expedition, 1925 to 27, with all these sections in the South Atlantic. And they always, every section, every station, they measured oxygen with the same method we can do today. And so we can compare these. And that's where we see these large changes, decline in oxygen. And we don't have that for temperature, where measurement protocols have changed, methods have changed quite a few times, and easily introducing errors, systematic errors of tens or few tenths of degree, even so a large part of the signal. So this is encouraging that oxygen is really, really a robust technique. We have been used, we've measured it for a long time, and we just have to collect all the data and all the information. And trying to do that here in this study is that the available measurements over time, the x-axis is time from 1900 to 2020, and we see there's quite some data in the second half of the last century. And as instruments change, particularly these glider measurements and automatic autonomous buoys came up and measured oxygen, the number increased. But again, they don't use Winkler, they use different methods. So we have to calibrate them carefully. We have to see if the instruments are robust. And so we have many additional issues to consider suddenly. And only the red bars here indicate the number of profiles of measurements, of profiles per year taken by Winkler. So that's the red part. And you see that is declining because less and less ships, research vessels are available. It's very expensive, of course. And we switch to other data sources, but these have to be calibrated carefully to extend these long-term observational asset that we have with oxygen data. And well, we have very uneven regional coverage of all these data. This is for each 5-year period over the last century. The coverage of the blue dots are these profiles where they are taken. So there are very few data in the beginning of the last century. Still a couple of data on the Southern Ocean, that's surprising. So that's where many of these international cruises went to. That was still terra incognita, and well, there was a hope to find valuable minerals or species for fishing. And then only after the Second World War, it really took off that we have almost global coverage every 5 years. But you see some decline in data coverage over the last years. This is due first to slow reporting and all this time people spent in doing other things, paper writing, proposal writing, and so on. But data quality control and data archiving is really essential, and we have to make sure that this is done for every single cruise, every single observations. It does enter the proper databases. To avoid this decline in observations in our databases, we have to make sure this is handled properly in all countries globally, and that's why this UN Ocean Decade is so important. That all participants, all countries really commit to making sure all the data obtained by their cruises on their shores and their national waters are reported, archived in accessible databases. Sorry. So coming back to what we need these data for. Well, first you saw these regional examples of low oxygen, declining oxygen, and also we need to find out, are we on track? Do we recover when we stop some action like putting in nutrients into the ocean or putting in CO2 into the atmosphere? So we need basically a proof of success if we want to, well, justify the amount of societal transformation, or which is will not be easy. It would be good to immediately or as quickly as possible show there's some success. So far, there's little success in oxygen. It's still declining. and we have these projections of models. But we have an issue there that all these current models are not very good in simulating past observed deoxygenation. So in fact, all these IPCC type models underestimate the observational estimate of the decline. So he wrote it's observed decline. It's still observation estimates because there are mapping issues. You have seen the uneven data coverage, uneven between years and between ocean basins, between seasons as well. So we have to make sure that we have a better data coverage and good interpolation schemes to really have a confident data observational estimate. But right now it looks like all our models severely underestimate the rate of oxygen loss. And this is alarming because oxygen is linked to both temperature, warming, but also to carbon because of respiration. And so if we want to get warming right and CO2 uptake right, we should get oxygen right in the ocean as well. And right now, it does look like we have a serious problem here, which we have to identify and to quantify, and therefore we need reliable data. And one instrument we have is the State of the Ocean report. And the last one just came out 2 weeks ago. It's by IOC and UNESCO. And it's available here. So every year, every other year, we have we describe the state of the ocean, warming, acidification, deoxygenation. And this is at least a call for action based on the best available information, scientific observations that we have. So it's similar like an IPCC report for climate. This is the state of the ocean. And you can look it up on this webpage. Or Google it, state of, or whatever search software you use, State of the Ocean Report 2024. So, but a concrete measure is the Global Ocean Oxygen Database and Atlas for assessing and predicting deoxygenation and also ensuring that our models are as good as they can be and should be. And that's the GO2NE project, GO2NE project here. Where we aim to collect all data, have quality control data, and have good interpolation schemes, mapping schemes to provide the necessary and robust, reliable information to society, to scientists, and to decision makers. How does it work? Well, we have data of different sources, World Ocean Database, and here's an example for the South Pacific. Then local, national data, individual cruises, Army and Navy data, some private organizations, very coastal communities that have want to check water quality. And all these sit often in different different databases, and combining these is a challenge. So we have done that here, and it was a hackathon event, very interesting, where early career people came together and brainstormed over a few days. What can be done? How can we combine these different data sources, access that data, make sure they have the same format, they have the same quality control, and then bring all these together into a single data set, quality control data set. And that's the idea behind this GO2NE project, where we aim to have develop an atlas of reliable oxygen information. And this is GO2NE is one example or one element of our decade program, Ocean Decade. That's the United Nations Decade of Ocean Science for Sustainable Development, this current decade. And the Global Ocean Oxygen Decade is one of its programs. There are many more programs, but we here run the Ocean Oxygen Decade program. The vision is to develop and, well, first find out and then do the science we need for the ocean we want. And that's an oxic ocean, we hope, and we want certainly. And the mission is to have to build and develop transformative ocean science. So that's not only by scientists, but with stakeholders in a joint operation where local knowledge is available from fishermen, local coastal people, environments that do have a lot of historical information, which is not in current databases and maybe not in scientific papers, but we have a lot of knowledge around that we want to combine in this transformative ocean science and connect the people and the ocean. What do we do in practice? Well, we do education capacity building, stakeholder engagement. This is an example of our last summer school last year in Chile, where we had, well, education program, but also a stakeholder program with local fishermen, fisherwomen. And they are really concerned about the oxygenation, and that hits, of course, their business. And they are also the people who know a lot of about past deoxygenation events and what you can do about it. Some adaptation, moving your nets, moving your lines for mussel farming, shellfish farming. So a couple of things you can do. And there's another thing, it's citizen science. That's a local project here around Kiel, northern Germany, and the Baltic, where we rent out these instruments to sailors that sail around here in summer for fun. But they can do some additional fun and provide data for science. And by putting these oxygen sensors into the water, taking profiles of oxygen, giving that instrument back in the next harbor, where it is then taken up by the next sailor. And that gives us a pretty good observation network, and very cheap, so it doesn't cost hours or days of scientists. It just costs the instruments, which are now pretty, well, some of these at least are relatively cheap already, and we can do that. We hope to do that in other regions of the world as well. And we have these, regional initiatives. So Baltic Sea was one example here, but we have one in the larger region, Western Pacific, Westpac program, where a number of working groups are very active to combine knowledge on ocean ecosystem health, on mariculture, on emerging oceanic issues. And it's really a regional network of training and research centers on ocean science. So this is a very promising example how regional networks can work very well. And we hope to set up more of these and encourage you as well to go ahead and set up more. Have win-win situations where you share knowledge, share information, and can develop solutions. Regional, because you have regional problems, which are maybe different in different parts of the ocean, in different parts of the world. But this is I think, encouraging way forward. So final slide, a call for action here. So what do we need in the global ocean oxygen decade, and what do we want to achieve or have achieved at the end of the decade? We want to have achieved increased knowledge about causes, impacts, and threats of deoxygenation. So that's quantitative knowledge, but also mechanistic understanding, and including its ecological, socioeconomic vulnerabilities, resilience, and of course also climate feedbacks. There are some greenhouse gases sensitive to redox, to oxygen levels in the ocean. And this is the more scientific understanding, knowledge, reliable knowledge. Second point, increased capacity to measure, document, and map and monitor, understand deoxygenation. So this is the data requirement. We have to do much better there. And from that, we can then, that's number 3, develop indicators or related methodologies that can be provided to agencies, industry, local communities to ensure safe operating spaces. This might even allow us to extend the concept of planetary boundaries and planetary health to marine oxygen. We will have a paper of our Global Ocean Oxygen Network out very soon on some ideas how to move forward this concept of extending planetary boundaries to ocean deoxygenation. So watch that space. It's in Nature, Ecology and Evolution coming out very, very soon. And then the last point is actionable strategies to mitigate deoxygenation. And if we cannot mitigate, to adapt to ocean deoxygenation on a local to global basis. So many things will be local because conditions are so different. And so we need this exchange with local stakeholders to have the local knowledge, but also local questions, of course, that may be very, very different in different parts of the world. But we all have to talk to each other and also invest in some science data and make sure the data and information is archived in the right way and is accessible, transparently accessible, free of any costs to anybody in the world. And that will hopefully increase and speed up knowledge generation. And this closes my presentation. Thank you very much for your attention. I look forward to any questions that you may have.
Okay, thank you very much, Andreas. We have time for questions, so so please, if you have questions, place them in the chat, in the Q&A, I should say. One question that has come up, Andreas, is about oxygen deoxygenation rates, I think, in North Africa. Is there anything you can say about that?
Yes, so yeah, we have pretty good data coverage there of West Africa, Northwest Africa, Cape Verde, around Mauritania, upwelling. And there's a pretty strong decline and expansion also. Well, overall an expansion of the anoxic or of the hypoxic waters. It's not yet anoxic, so we have several 30, 40 micromolar oxygen concentrations in most places offshore. But we do see a decline, and this is pretty well covered. There were many observational campaigns over the last few decades. But we have seen a steady decline in the lowest oxygen concentrations in that area. It is modulated a little bit by equatorial current bands and that can shift interannually. There's interannual variability, interannual modes, climate modes that do control how much oxygen is coming from the Western Atlantic towards Africa via these zonal current bands from the west to the east. So that could does change ventilation a little bit, but overall there's a strong decline from about, I think, 50 micromolar lowest levels some 30 years ago to about 40 or less, even less than 40, 35, I think around is the current value micromole in the last current decade.
Thank you.
And it might be the next, really anoxic region developing there that we can see during maybe our lifetime even. And so that's really a concern that the Atlantic might become like the Pacific, where we do have anoxic waters in the upwelling regions.
Okay, there's another question about pumping oxygen or pumping air into the ocean at the seashore and deeper in the sea. Is it worth doing that to improve the oxygen level in the sea?
Yeah, that's a good question. And there's right now many green hydrogen companies and initiatives that produce hydrogen, but also have a waste product of oxygen, and they would be happy to use it for some good purposes, and maybe also increase their financial benefit. But there's, yeah, so there's some oxygen available. And the idea that has been done in lakes quite a few times to ventilate, pump oxygen into lakes to avoid anoxia in lakes. In the ocean, it's very difficult. So the global ocean, we don't have enough oxygen in all these hydrogen electrolyzers. So that is a minuscule amount. So it would not help to fight global deoxygenation. Locally, it might be possible in some bays, so some enclosed bays with very low volume of anoxia. They could be targeted, I think, with pumping air or oxygen into it. The question is, do you really, or could you do more good than do harm to the system? It depends how you pump. If you bubble it, you may completely mix it. You do mechanical harm to some organisms. You often pump, or there have been some trials that pumped in surface waters, oxygenated waters down to deeper levels. So then you change temperature profile stratification. And I think two out of three cases I'm aware of in the Baltic Sea, where that has been done some 15 or 20 years ago, two of these failed because they pumped down oxygen, but they also pumped down warmer waters that increased respiration at depth. And in the end, even though they pumped down oxygen, they had increased respiration at depth, and in the end, lower oxygen levels. So they increased the stress, the oxygen stress. And so there are many of these feedbacks that we have to take into account very carefully. And there's also these, what you mentioned in your talk, Caroline, all these microbial processes in the sediments that may release some toxic elements. They may release may increase respiration in the sediments as well, and thereby consume most of the oxygen, release CO2 or other greenhouse gases. And so we may possibly have a decline in anoxic waters, but at the expense of producing more greenhouse gases. And so that has to be evaluated very carefully, I think. It might work in some cases, but we have to do that. And that's also some of the things the Global Ocean Oxygen Network might, or will we want to evaluate that more carefully to give better advice.
Yeah, there's a related question about denitrification, that if you pump in oxygen, that your denitrification will become less, and that you will then could get more organic matter production. And the question there is if you know of any methods to reduce the organic matter production, then to avoid the amplification of deoxygenation. That's
not sure. Well, you could basically you have to take it out and store it safely somewhere. So you don't want, well, killing the ocean or killing biology wouldn't help, since it's also producing oxygen, of course, and it produces as much as it consumes. So you really would have to take out organic carbon, organic matter that is already somewhere in the ocean. You have to fish it or condense it somewhere and lock it away. And that's pretty difficult. You could try to sink it to great depth, so compress it and sink it, so to move it away from well, upper ocean oxygen at least. No, but I could also,
yeah, I could also just add that from a systems perspective, that making the biotic system more whole, I think, can actually have an impact. So I'm thinking about things like restoring beds of oysters, for example, which or menhaden types of fishes that actually short circuit the sort of trophic dynamics and directly feed on phytoplankton, can actually take up an awful lot of this organic matter production and prevent the deoxygenation. In fact, I think mussel farming has been suggested as one measure to sequester nutrients and organic matter in Sweden, for example.
Yeah, thanks. Yeah, shorten the food web and reduce respiration.
okay. So there's also a question about the impact of ocean deoxygenation on seaweeds, but maybe you can, or make that vegetation, or yeah, I'm not sure we have the expertise there, but maybe you can comment.
Yeah, no, I'm not the expert there, but I guess, well, they are autotrophic and they produce their own oxygen. They do not depend on it, but they, of course, they have symbionts that are heterotrophic and that do depend on oxygen. So they are entire ecosystems, and they are probably, and well, hydrogen sulfide should not be present. That's it will even kill many autotrophic, or maybe all autotrophic organisms. I don't know. So anoxia really could be harmful even to autotrophs, to oxygen producers.
There's a question about past greenhouse events, which is quite a complicated one, but it says that denitrification may have decreased during past greenhouse events, implying that OMZs actually decreased during warming. Do you have any thoughts on how to explain this? This might be a rather challenging question. That's what the person says, but maybe, yeah.
Yes, it is. I don't know. Well, ocean ventilation is certainly the answer there, and that's really the critical point now. If we look at the past 500 million years of the planet, continental configurations have changed quite a bit, and we do know that is a major control on ocean ventilation and on ocean anoxia. It's not oxygen in the atmosphere. It's not primary production. It's to some extent nutrient input into the ocean, but a large portion is ocean circulation. And you can have circulations even in warm climates that are very rapid. And well, in fact, if you take today's ocean and just increase the temperature everywhere by 3 degrees, the ocean will turn over faster. So that's the dynamics is, or the state equation pressure gradients will be so that the ocean will ventilate faster in a warmer climate. And we see that even right now in our models. So if we integrate them for a few thousand years into the future, we have more oxygen in the ocean. In a warmer ocean, that can happen. But still we see that the anoxic areas, they do expand. And so these shadow zones also expand, and it's just a different distribution. But that's ocean dynamics mostly, I think. And that if you, depending on where your cores, where your sediment archives are from, you may be in an area where you see an increase in oxygen in warmer climates. And that is very well possible because circulation usually is faster at higher temperatures.
Okay, thank you. I think it's time to wrap up. So yeah, thank you to both speakers, so Karin and Andreas, and thank you for your answers to the questions. Before we go, I want to actually show 2 quick slides. I hope you can see this, because I'd like to just highlight what GO2NE and GO2NE do. There's an ocean oxygen website that has links to past webinars. So also this webinar will be available there. And there's also a good news newsletter on ocean deoxygenation. And there's a feed with a lot of oxygen, or the latest scientific articles on ocean deoxygenation. So please check out the website. And you can also find links to IOC, UNESCO, and there's a lot of information there on their activities on those web pages. And with that, I want to thank the speakers again, and thank you for being here today for this presentation. The next webinar will be in July, and in the meantime, you can follow the posts on social media, and I hope you have a pleasant day or night wherever you are. And so thank you for being here. Goodbye.